Hello students, welcome to EduFarm. Believe and Become classes for GPAT and NIPER-JEE. Like, share, subscribe to our channel, hit the bell icon to get further notifications. Hello students, welcome to another video by EduFarm. Hope you all are doing well. Today's topic is on carbohydrates. This is a part from the Biochemistry section of the syllabus for GPAT, NIPER as well as for JELET and semester exams. This part is also important for NEET exams. Now let's begin without any ado. Now what are carbohydrates? Carbohydrates are polyhydroxy aldehydes or ketones or substances that yield such compounds on hydrolysis. Many but not all carbohydrates have the empirical formula of CH2O(N). Some also contain nitrogen, phosphorus or sulfur. Now carbohydrates can be classified into monosaccharides, also known as simple sugars, as it consists of a single polyhydroxy aldehyde or ketone unit. For example, D-glucose or dextrose. Then oligosaccharides. In oligosaccharides we have mentioned disaccharides also. So disaccharides and oligosaccharides become one part. These are short chains of monosaccharide units or residues joined by glycosidic bonds. Example sucrose or cane sugar, which consists of 6 carbon sugars, D-glucose and D-fructose. All common monosaccharides and disaccharides have names ending with the suffix "ose". Now monosaccharides have 1 unit and disaccharides have 2 units and oligosaccharides have 3 to 10 units of monosaccharides. Now the third one is polysaccharides. Sugar polymers containing more than 20 or so monosaccharide units, and some have hundreds or thousands of units also. Examples are cellulose for linear chain polysaccharides and glycogen for branched chain. Now here is an example of D-glucose, which consists of 1, 2, 3, 4, 5 and 6 carbons. Here is an example of sucrose, which is made up of glucose and fructose units and is joined by the glycosidic bond. Now here is the example of glycogen. Now glycogen is made up of a structure like this. Here we can see the bonds between 1 and 4th carbon of 2 units and 1 and 6 carbon of 2 units joined by oxygen. Now these sites are the non-reducing agents, and on the right side we have the R group, which shows that more alkyl groups can be added to that part. Now between 1 and 6 it is known as the alpha 1,6 glycosidic bond, and between 1 and 4 it is known as the alpha 1,4 glycosidic bond. Now monosaccharides. The number of carbon atoms on this basis and the functional groups present. Monosaccharides can be classified, and the examples are for trioses, that is which have carbons 3 in number, aldoses are glyceraldehyde, and ketoses are DH. Aldoses have the functional group of aldehyde and ketoses have the functional carbonyl group. Now for tetroses we have erythrose and erythrulose for ketoses. Pentoses, on aldoses we have ribose, then ribulose for ketoses. Similarly for hexoses, which has carbon number 6, it has glucose and fructose. As well as for heptoses, which have a carbon number of 7, the aldoses have glucoheptose, and for ketoses we have pseudoheptose. Now what are some of the tests by which we can detect the presence of monosaccharides or carbohydrates? in a sample. By Molish test, we can detect carbohydrates. By Benedict test, we can detect the reducing sugars present in the sample. By Barfoed's test, we can detect the monosaccharides. And by Sullivan's test, we can detect the ketosis present. Now disaccharides. Disaccharides are classified into two types, reducing and non-reducing. Now in reducing, we have lactose, which has a beta 1,4-glycosidic link and is made up of two units of one of galactose and one of glucose. Again we have maltose, which has an alpha 1,4-glycosidic linkage and is formed by two units of glucose. In non-reducing part, we have one and only alpha 1 beta 2 glycosidic linkage containing sucrose, which is made up of glucose and fructose. Then comes oligosaccharides. Oligosaccharides are examples are rhamnose, stachyose, and raffinose. Now in polysaccharides, we have homopolysaccharides and heteropolysaccharides. Homopolysaccharides means it has the same sugar units, and heteropolysaccharide means it is made up of different sugar units. In homopolysaccharides, we have starch, glycogen, cellulose, dextrin, dextran, chitin, inulin, etc. And in heteropolysaccharides, we have agar, agarose, hyaluronic acid, heparin, chondroitin sulfate, keratan sulfate, dermatan sulfate. In starch, we have two types of glycosidic bond, that is alpha 1,4-glycosidic bond and alpha 1,6 glycosidic. Glycogen, as we know, is used as the storage carbohydrate in animals. Then comes cellulose, which has a beta 1,4-glycosidic linkage. Cellulose is also helpful in obesity, diabetes, and cardiovascular disorders. Then we have dextrin. Dextran is also an example of homopolysaccharides, which is a plasma expander. Then we have chitin, which forms the exoskeleton of many insects. Then inulin, which is a polymer of fructose and is used in the estimation of glomerular filtration defects. In the heteropolysaccharides, we have agar, agarose, and as I said, hyaluronic acid, heparin, and these are all known as glucose aminoglycans or mucopolysaccharides. These are mucus in nature and anionic in charge. Due to the negative charge present, they stick together and form a mucus-like or sticky substance. Now the functions. Now glucose is used in energy as an energy source to provide 4 kilocalorie per gram energy, and it is used as an energy source for brain and red blood cells. Then we have sucrose, which is used as cane sugar. Then we have lactose, which is used as milk sugar to provide nutrition to the newborn babies. Then starch, which is used as a storage carbohydrate in plant. Glycogen is a storage carbohydrate in animals. Hyaluronic acid is a shock absorber present in synovial fluid and the vitreous humor of our eyes. Then we have heparin, which is used as an anticoagulant, and dextran, which is used as a plasma expander. Now more functions are: the carbohydrates are supply carbon atoms for the synthesis of biochemical components like nucleic acids, lipids, and proteins. They form the structural framework of DNA and RNA. Glycoprotein and glycolipids form component of the cell membrane and receptor. Chitin forms the exoskeleton in insects. Carbohydrates help in preventing ketosis. They help in peristaltic movement of food, as well as they help in secretion of digestive juices in the GI tract. Now, for the digestion of carbohydrates, the carbohydrate-rich food is intake to the mouth, and then it is considered starch in food. Now, enzyme alpha amylase present in the salivary enzymes cause the hydrolysis of alpha-1,4-glycosidic bonds. Then what happens? The partially digested starch, that is oligosaccharides and shorter polysaccharides, are transferred to the stomach, where the salivary amylase gets deactivated due to the presence of stomach acids, and then these partially digested starch travel to the duodenum, and in the lumen of the duodenum an enzyme alpha amylase, which is again secreted by the pancreas, so this is known as the pancreatic alpha amylase, again causes the hydrolysis of alpha-1,4-glycosidic bonds. Then what happens? These units travel to the intestine, where the brush border epithelium or the enterocytes release brush border glucosidases. These are maltase, sucrase or isomaltase, alpha glucosidase. These finally convert all the partially digested starch into smaller glucose units, and which are ready for absorption. Now in case of absorption, in our lumen the glucose units are present with many sodium ions. Now in the enterocytes we can see these sodium glucose link transporters. These sodium glucose link transporters help to enter the sodium and glucose units into the enterocytes by sodium glucose link transporters. For every 1 glucose unit, 2 sodium units enter the enterocytes. Then again on the basal side, as we have the sodium glucose link transporters on the apical side, on the basal side we have GLUT2 receptors, which further transfers or absorbs the glucose units into the blood, and from here it is used as an energy source and stored as glycogen. Now some more starch remains undigested. These rest of the undigested starch, which are also known as resistant starch, is fermented by the bacteria of the gut into short chain fatty acids and absorbed by the body. Now this is all for today's video. We will move on to the next video with further topics regarding carbohydrates. Hope you all are well in this pandemic and keeping your preparations high. Keep preparing, keep growing. Thank you to watch this video. See you in the next video.